enormously, continuously.
So each testis is an organ suspended within a pouch of skin known as the scrotum. The testes thus lie outside the abdominal cavity, and this ensures that spermatogenesis, the formation of spermatozoa, occurs at the temperature slightly below body temperature, a requirement for successful gamete production. Each testis is surrounded by a tough connective tissue covering, known as the tunica albuginea. Connective tissue septa from the tunica albuginea penetrate into the substance of each testis and divide it into lobules. One septum forms a wedge which penetrates the posterior surface of each testis. Located within the lobules of each testis are coiled seminiferous tubules, which are the sites of spermatogenesis. Seminiferous tubules are lined by several layers of epithelial cells, forming a germinal epithelium. There are two types of cells in the germinal epithelium of adults. First is spermatogenic cells, which are in layers, and second is Sertoli cells, which form a single layer and are supportive of spermatogenic cells. The germinal epithelium lies on basement membrane and is surrounded by a thin layer of loose connective tissue. This connective tissue supports blood vessels, lymph capillaries, nerves, and Leydig interstitial cells, which secrete androgens. Myoid cells are closely adjacent to seminiferous tubules and are capable of contraction, thus helping to move spermatozoa along tubules and away from where they develop. After puberty, spermatogenesis begins as spermatogenic cells undergo meiosis and differentiation in a cycle of events that result in production of spermatozoa, a process which takes about 65 days in humans. The stages of spermatogenesis take different lengths of time to complete, and as a result, not all the stages are present in layers of cells in any one region of germinal epithelium. The appearance of seminiferous tubules depend on the stage the spermatogenic cells have reached in forming spermatozoa and on the angle at which they are sectioned. The formation of spermatozoa, spermatogenesis, may divide it into three stages: mitosis, meiosis, and spermiogenesis. Those stages begin at about puberty and continue throughout life. In mitosis stage, stem cells, spermatogonia, at the base of the germinal epithelium undergo regular cycles of mitosis and produce new diploid cells, that is, cells containing the normal number of chromosomes. Some of the new cells formed by mitosis remain in the basal layer as spermatogonia, others move towards the lumen and enter meiosis. At the start of meiosis phase, the cells begin to enlarge and are known as primary spermatocytes. They undergo an S phase, then they replicate the DNA in their chromosomes prior to cell division. They then enter the prophase of the first division in meiosis, a process which lasts about 20 days. During this time, chromosomes begin to coil and condense and stain densely, and homologous pairs of chromosomes move close together and exchange lengths of DNA. This exchange introduces genetic variation to the gametes produced. The first meiotic division of meiosis, also known as the reduction division, continues into metaphase. Each pair of homologous chromosomes in each primary spermatocyte line up together on the same part of the equator of the spindle of the metaphase plate, prior to cell division. This contrasts with mitosis, where each chromosome attached to a different part of the equator of the spindle. At this stage, the chromosomes are at their most condensed and densely stained, and are rapidly identified in histological sections. The paired chromosomes then move away from each other in anaphase and telophase. The result of this reduction division is the production of two secondary spermatocytes, each with only half the original number of chromosomes. This is 23 in humans. This reduction division introduces further genetic variation to the new cells, as maternally and paternally derived chromosomes in the homologous pairs are randomly arranged on the spindle, and therefore randomly distributed between the two offspring cells. The second meiotic division, known as the meiotic division of meiosis, occurs rapidly as chromosomes in secondary spermatocytes line up as individuals, as they do in mitosis, on the metaphase spindle of this division. The chromosomes then split along their length and separate into two new cells, known as spermatids. As this cell division is rapid, few cells in any one histological section are in this stage. Each resulting spermatid has 23 chromosomes with the haploid amount of DNA. This is half the number of chromosomes and half the DNA in the stem cells, spermatogonia, and of most other cells in the body. Separation of cytoplasm during meiosis is incomplete, and spermatids derived from one spermatogonium are partially joined together. This is thought to aid synchrony of their progression through the next stage, spermatogenesis. Groups of newly formed related spermatids appears as clusters of small, spherical cells near the luminous surface of seminiferous tubules. During spermatogenesis stage, spermatids differentiate and each form one spermatozoon. Approximately 200 million spermatozoa are produced daily by the testis in humans. During spermatogenesis, the spherical spermatids change in form and shed cytoplasm as they become highly differentiated spermatozoa characterized by a head, middle region, and tail. As spermatozoa develop, the head region remains the epithelium, but the tail region extends into the lumen of the seminiferous tubule. Eventually, spermatozoa pass into the lumen, and fluid secreted by Sertoli cells aid their transport away from the seminiferous tubules. The head of each spermatozoon contains the nucleus in which the DNA, in the chromosomes, is condensed and strains densely with bases such as hematoxylin. At one end of the nucleus of developing spermatozoon, a head, known as a cap, develops known as the acrosome. The contents of the acrosome include hydrolytic enzymes that take part in acrosome reaction, which is the process by which the head of the spermatozoon penetrates the cells and structure surrounding the oocyte prior to fertilization. The middle region of spermatozoon is mainly occupied by mitochondria, which provide energy for the movement of the tail. The tail has a 9+2 arrangement of microtubules characteristic of cilia, and those provide the motive force which allows spermatozoa to swim. However, spermatozoa become self-propelled only after they have left the testis and are fully matured. Then they are then able to undertake unidirectional travel at about 3 mm by minute. Sertoli cells are tall and columnar, and extend from the base of germinal epithelium to the lumen of the seminiferous tubule. The nucleus of Sertoli cells may appear oval or triangular, and most display a prominent nucleolus and palely stained chromatin, reflecting their high level of synthetic activity. Developing spermatogenetic cells are enfolded in the lateral cell membranes of Sertoli cells, and as a result, detail of Sertoli cell structure is difficult to distinguish in routine histological section. Adjacent Sertoli cells are joined to each other near to the basement membrane by tight junctions. This arrangement separates spermatogonia into basal compartment of germinal epithelium and the developing spermatogenic cells from primary spermatocyte to spermatozoa into an adluminal compartment. The barrier formed between the two compartments is known as the blood-testis barrier, and it ensures that large molecules, for example antibodies or microorganisms in blood, do not readily pass to developing spermatozoa. Sertoli cells are also important in nourishing developing spermatogenic cells, and they phagocytose cytoplasmic fragments released from spermatozoa as they develop. An important component in the secretion from Sertoli cells is an androgen-binding protein. This binds testosterone and helps to ensure levels in the seminal tubules are higher than in the circulation, and the level is adequate from development of spermatozoa. In addition, fluid secreted by Sertoli cells helps to move spermatozoa away from the testis, as at this stage the spermatozoa are not able to propel themselves. Leydig cells are endocrine cells in the loose connective tissue around seminiferous tubules, and they secrete mainly testosterone. The Leydig cells are stimulated to produce and secrete testosterone by a luteinizing hormone secreted by cells in the anterior pituitary gland. Testosterone enters the circulation and stimulates secondary sexual characteristics, including skeletal muscle development and the patterns of hair growth. However, circulation levels of testosterone are insufficient to maintain spermatogenesis. Testosterone levels in the testes are kept high enough to stimulate spermatogenesis as the androgen-binding protein secreted by fertility cells bind testosterone in the testes. Another hormone from anterior pituitary cells, follicle-stimulating hormone, stimulates fertility cells to secrete the androgen-binding protein. Most seminiferous tubules are coiled loops which have a strain region, tubule recti, at each end, through which fluid and spermatozoa live. The tubule recti drain into the meshwork of spaces lined by a cuboidal epithelium known as the rete testis. The rete is embedded in the vegi of connective tissue which extend into each testis from the tunica albuginea. The contents of the rete testis is drained into 12 to 15 ductal efferents, which is turned drain into a single duct in epididymis. Each epididymis is formed by the ductal efferents and the ductus epididymis. The 10 to 20 ductal efferentes are short tubules that drain spermatozoa from the rete testis and pierce the tunica albuginea to conduct the sperm to the ductus epididymis. Thus, the ductal efferents become confluent with the ductus epididymis at this point. The simple epithelial lining of the lumen of each ductule consists of pouches of non-ciliated cuboidal cells altering with region of ciliated columnar cells. The successive clusters of short and tall epithelial cells imparts a scalloped appearance to the lumina of the ductule efferentes, a notable characteristic of those ductules. The cuboidal cells are richly endowed with lysosomes, and their apical plasmalam display numerous invagination indicative of endocytosis. Those cells are believed to resorb most of luminal fluid elaborated by the Sertoli cells of the seminiferous tubule. The cilia of the columnar cells probably move the spermatozoa towards the epididymis. The simple epithelium sits on a basal lamina that separates it from the thin loose connective tissue wall of each ductule. This connective tissue is surrounded by a thin layer of smooth muscle whose cells are circularly arrayed and by undergoing rhythmic contraction assisting properly in their luminal content into the epididymis. The ductus epididymis is a thin, long, 4 to 6 meters in length, highly convoluted tubule that is folded into a space only 5 cm long on the posterior aspect of the testis. The epididymis may be subdivided into 3 regions: head, body, and tail. The head, formed by the union of the 10 to 20 ductal efferents, become highly coiled and continues as the equally highly coiled body. The distal portion of the tail, which stores spermatozoa for a short time, loses its convolution as it becomes continuous with the ductus deferens. The lumen of the epididymis is lined by a pseudo-stratified epithelium composed of 2 type cells. First is short basal cells. They are pyramidal to polyhedral. They have round nuclei in which large accumulation of heterochromatin impart of a dense appearance to the structure. The sparse cytoplasm of those cells is relatively clear with a scarcity of organelles. It is believed that the basal cells function as stem cells, as well as the principal cells as the need arises. And second type the tall principal cells. They have irregular, oval nuclei with 1 or 2 large nucleoli. Those nucleoli are much paler than those of the basal cells and are located basally within the cell. The cytoplasm of principal cell houses abundant rare located between the nucleus and the basal plasmalemma. The cytoplasm also has a large supranuclear position Golgi complex, numerous profiles of apically located smooth endoplasmic reticulum, endoplasmasomes, and multivesicular bodies. The apical cells membranes of the principal cells display a profusion of pinocytotic and coated vesicles at the basis of the many stereocilia that project into the lumen of the epididymis. Those long branchy cellular extensions are clusters of non-motile microvilli that appears to form clumps as they adhere to one another. The principal cells resorb the luminal fluid, which is endocytosed by pinocytotic vessels, and delivered to the endolysosomes for disposal. Additionally, those cells phagocytose remain of cytoplasm that were not removed by Sertoli cells. Principal cells also manufacture glycerophosphochelin that inhibits spermatozoon capacitation, thus preventing the spermatozoon from fertilizing a secondary oocyte until the sperm enters the female genital tract. Additionally, the high level of fertilization-promoting peptide also prevents the spermatozoon from undergoing capacitation until it reaches the female reproductive tract, where FPP levels become diluted. The epithelium of epididymis is separated by the basal lamina from the underlying loose connective tissue. A layer of circularly arranged smooth muscle cells surrounds the connective tissue layer. Peristaltic contraction of this layer helps conduct the spermatozoa to the ductus deferens. Each ductus deferens is a thick-walled muscular tube with small irregular lumen that conveys spermatozoa from the tail of the epididymis to the ejaculatory duct. The stereociliated pseudostratified columnar epithelium of the ductus deferens is similar to that of the epididymis. Also, the principal cells are shorter. A basal lamina separates the epithelium from the underlying loose fibroelastic connective tissue, which has numeral folds, thus making the lumen appear irregular. The thick smooth muscle coat surrounding the connective tissue is composed of three layers: inner and outer longitudinal layers with an intervening middle circular layer. The smooth muscle coat is invested by a thin layer of loose fibroelastic connective tissue. The dilated terminus of each ductus deferens, known as the ampulla, has a highly folded, thickened epithelium. As the ampulla approaches the prostate gland, it is joined by the seminal vesicle. The continuation of the junction of the ampulla with the seminal vesicle is called the ejaculatory duct. Each ejaculatory duct is a short straight tubule that enters the substance and is surrounded by the prostate gland. The ejaculatory duct ends as it pierces the posterior aspect of the prostatic urethra at the colliculus seminalis. The lumen of the ejaculatory duct is lined by a simple columnar epithelium. The subepithelial connective tissue is folded, a feature responsible for the irregular appearance of its lumen. The ejaculatory duct has no smooth muscle in its wall. The paired seminal vesicles are highly coiled tubular structure about 15 cm long. They are located between the posterior aspect of the neck of the bladder and the prostate gland, and join the ampulla of the ductus deferens just above the prostate gland. The mucosa of seminal vesicles is highly convoluted, forming labyrinth-like cul-de-sacs that in 3 dimensions are observed to open in central lumen. The lumen is lined by a pseudostratified columnar epithelium composed of short basal cells and low columnar cells. Each columnar cell has numerous short microvilli and a single flagellum projecting into the lumen of the gland. The height of cells varies directly with blood testosterone levels. The subepithelial connective tissue is fibroelastic and surrounded by smooth muscle cells, arranged as inner circular layer and outer longitudinal layer. The seminal vesicle once were believed to store spermatozoa, some of which are always present in the lumen of this gland. It is now known that those glands produce a yellow, viscous, fructose-rich seminal fluid that constitutes 60% of the volume of semen. Also, seminal fluids also contain amino acids, citrates, prostaglandins, and proteins. Fructose is its principal constituent because it is the source of energy for spermatozoa. The characteristic pale yellow color of semen is due to the lipohormone pigment released by seminal vesicles. The prostate gland, the largest of the accessory glands, is pierced by the urethra and the ejaculatory ducts. The slender capsule of the gland is composed of richly vascularized, dense, irregular collagenous connective tissue interpressed with smooth muscle cells. The connective tissue stroma of the gland is derived from the capsule and is, therefore, also enriched by smooth muscle fibers, in addition to their normal connective tissue cells. The prostate gland, a conglomeration of 30 to 50 individual compound tubular acinar glands, is arranged in 3 discrete concentric layers: mucosal, submucosal, and main. Each tubular-acinar gland has its own duct that delivers the secretory product into the prostatic urethra. The mucosal glands are closest to the urethra and thus are the shortest of the glands. The submucosal glands are peripheral to the mucosal glands and are consequently larger than the mucosal glands. The largest and most numerous of the glands are the peripheral, most main glands, which compose the bulk of the prostate. The components of the prostate gland are lined by a simple to pseudo-stratified columnar epithelium, the cells of which are well endowed with organelles responsible for the synthesis and packaging of proteins. The lumina of the tubular-acinar glands frequently house round to oval prostatic corpora amyloidea, composed of calcified glycoproteins whose numbers increase with a person's age. The bulbourethral Cooper's gland is a pear structure about 0.5 cm in size, located on either side of the urethra in the urogenital diaphragm below the prostate. A bulbourethral gland is typically made of tubules and acini, which is why we can characterize it as a tubular-alveolar gland. Each gland is formed of numerous of lobules that are enclosed with the fibrous capsule. Histologically, they are lined by pseudo-stratified epithelium. Within those lobules, we find acini lined by columnar cells. A thin layer of mesoepithelial cells surrounds each separate acini. Microscopic analysis of those cells show a large amount of intracytoplasmic mucin. Each of those acini open into a duct, which joins other adjacent ducts, and all eventually form the central excretory duct of the gland. Cuboidal cells line each duct. The penis is composed of 3 columns of erectile tissue, each enclosed by its own dense fibrous connective tissue capsule, the tunica albuginea. 2 of those columns of erectile tissue, the corpora cavernosa, are positioned dorsally. Their tunica albuginea are discontinuous in place, permitting continuation between their erectile tissues. The 3rd column of erectile tissue, the corpus spongiosum, is positioned ventrally. Because the corpus spongiosum houses the pineal portion of the urethra, it is also called the corpus cavernosum urethra. The corpus spongiosum ends distally in the enlarged, bulbous portion, the glans penis. The tip of the glans penis is pierced by the end of the urethra at the vertical slit. The urethra lined with the pseudo-stratified columnar epithelium. The mucous glands of litera and ducts secrete a clear mucus on erection. The 3 corpora are surrounded by a common loose connective tissue shed, but no hypodermis, and are covered by a thin skin. The skin on the proximal portion of the penis has coarse pubic hair and numerous sweat and sebaceous glands. The distal portion of the penis is hairless and has only a few sweat glands. Skin continues distal to the glans penis to form a retractable shed, the prepuce, which is lined by a mucous membrane, a moist, stratified squamous, non-keratinized epithelium. Then an individual is circumcised, it is the prepuce that removed. Erectile tissue of the penis contains numerous variably shaped endothelially lined spaces that are separated from one another by a trabeculae of connective tissue and smooth muscle cells. The vascular spaces of the corpora cavernosa are larger centrally and smaller peripherally, near the tunica albuginea. However, the vascular spaces of the corpus spongiosum are similar in size throughout its extent. The trabeculae of the corpus spongiosum contain more elastic fibers and fewer smooth muscle cells than those of the corpora cavernosa.